My name is M Haberman. I'm the director of the Flow Cytometry Core. This is another fee-for-service facility that's open to all School of Medicine investigators. It is expertly managed by Jeff Lyon, who is sitting over there at this table. Okay, so flow cytometry is now a fairly standard technique that is employed by virtually every lab on this campus. As you're likely aware, it can be used to quantify multiple aspects of a cell, including its cell size, complexity of its intracellular compartment. These characteristics are often very indicative of particular cell types. And of course, any fluorescence associated with a cell can be quantified at a population level. So this facility is comprised of a very large collection of flow cytometers, cell sorters. However, many people are not aware that we also oversee a Biorad bioplex, which is used for Luminex samples, and an Amnis image stream imaging cytometer that I'd like to talk a little bit more about at the end. Many of these instruments have special features and advantages over the other ones that is unlikely to be discerned even from our website. Please, if you have any specialized interest, don't hesitate to contact any of our staff who can help you choose the appropriate instrument for your particular needs. Special options that are available that many people are unaware of as well include a biosafety containment for some of the sorters for particularly hazardous or high-risk materials. There is also the option for temperature control. Aside from the ability to ice your samples during collection, you can also heat them for those people who need to both sort and collect at elevated temperatures, for example. Plate and slide deposition is also possible, as well as single cell and plate index sorting. This is actually fairly powerful for people who have to do complex and calibrated population studies. One of the sorters, the Sony, aside from being in a biosafety cabinet, also has the feature of being double-headed. So those individuals who need to sort through a very large quantity of material very rapidly might want to employ this sorter instead. And finally, I'd like to mention a couple things about the Stratodyne flow cytometer. One of them is outfitted with a plate loader, which many people find very helpful, but it's also exceptionally low noise. So an individual who might be interested in, say, a very small material, say on the size of bacteria, might find their analysis better suited on the Stratodynes than a more classic LSR2. This is somewhat shifted. So for historic reasons, the vast majority of the equipment is currently in the TAC building. And that's in part because the original consolidation of this equipment was motivated by immunobiology staff. But as this campus has grown and lines of research have expanded in more disparate areas, so has the placement of equipment. So now some of our more highly configured, better equipment is actually placed in the Amistad building or at 300 George. Here originally to support the Stem Cell Center and Human Translation and Immunology. But as is the case with all of the equipment on this campus that we oversee, every instrument that we manage is available to all investigators. There's also a cell sorter inside the biosafety level 3 facility. Anybody who's contemplating large quantities of unscreened human material, we would prefer at the moment for that to be done inside a biosafety cabinet. We are undergoing an initiative to acquire a biosafety cabinet for the Amistad cell sorter as well. Okay, so this is a fee-for-service facility, but the emphasis here, I think, really is on the service. We have a lot of very talented, extremely talented staff, any of whom you can approach directly, or of course you can write to me or the facility manager, Jeff Lyon. Most of our staff operates cell sorters, and as is the case with Zhao and Susanna, they are dedicated to a particular sorter and an affiliated LSR2. But Jeff Lyon and Gazelle are switch hitters and actually operate more than one instrument on an as-needed basis. We've recently added an additional staff member, Christine Cody, who does exclusively support for the analyzers. And this has freed up some of the time for Eva Mennett, who now can spend some of her time operating a sorter as well. So even longtime investigators here at Yale may not appreciate that there are pairs of analyzers and sorters, not necessarily next to each other, but similarly configured, or at least similarly outfitted with lasers. And the idea here is that you should be able to do your preliminary studies on LSR2 and feel very confident in moving that same style of gating, that same panel of antibodies onto a sorter. So this is something that is available information on the website. At this moment, they're not exactly identical in all their configurations because we've recently upgraded the emission bandpass and the detectors on the analyzers to take advantage of some newly emergent dyes. And the sorters haven't yet caught up on that, but we hope to rectify that soon. So of course, there's a very wide range of fluorophores that can be detected with each of these instruments. And this can be confusing for a lot of individuals, but please don't allow that to be intimidating or to press the envelope in your particular panel design. Don't hesitate to contact one of our staff to get advice on what might be the best instrument for you. In addition, I wanted to mention that we have a computer workstation on the 6th floor in the TAC building where you can do FlowJo or CellQuest analysis or to further analyze the MNIST imaging cytometer data. Okay, so with the remaining time, I'd like to spend a minute talking a little bit more about imaging cytometry. We feel that this instrument, which is extremely powerful in what it can deliver, is underappreciated at the moment. So this is actually a hybrid between a microscope and a flow cytometer. Cells that are stained are sent through fluidics just like it would be for flow cytometry, but in addition, images are acquired of each cell as it passes by. So given that, it's actually fairly impressive that it still can screen 5,000 cells per second with real-time compensation. That is to say that as they are moving, you're able to observe exemplary images of your analyzed population, even with gating. It's fairly well configured, so that allows for quite a number of colors, but if you're interested in pursuing this technology, it's very important that you contact staff because the fluorophore selection is extremely critical. Compensation with image analysis is really something very different with flow cytometry, and you really don't want to be heavy-handed in any channel overlap in this regard. There's very strong support for this instrument, which right now is partially subsidized by a rheumatology P30. So Christine is there to hold your hand for not only for the training, the data acquisition, and the image analysis, but substantially thereafter to make this a really smooth process for you. The image analysis is very sophisticated, but still quite intuitive, that incorporates very traditional flow cytometry gating. And I'll show you this with a couple of examples in just a minute. But here, looking just at this very broad overview of some of the type of images you might get, you can see very quickly how some features of flow cytometry are insufficient for what you would like to quantify. Really at a very broad population-based level in the way that standard, say, confocal image analysis may not get for you. For example, if internalization was important, you could use this approach through the image analysis to quantify the amount of fluorescence, say, here in your green channel at the membrane, as opposed to what is internal to that cell, and even quantify the number of individual spots within that, its mean fluorescent intensity or its colocalization properties. Cell signaling is where it actually has a particular strength, Any sample that is also stained with any kind of a nuclear dye, again through image analysis, you can identify with a bright field overlap the edges of the cell eroding in what is cytosolic, masking just instead to your DNA binding dye, the edges of the nuclei, and there within your nuclear mask to be able to identify the amount of fluorescence within a channel. And as you can imagine, if you were hoping to observe a shift from the cytosol to the nucleus, here you can create ratios and plot even your image analysis parameters on your axes. Let's see just a couple of examples here. This here is showing a cell cycle measurement using the image stream imaging cytometer. You can plot your flow cytometry data on X or Y, and then using standard gating style, FlowJo style, be able to select a subset. Each event then is something that you can observe as an independent image, giving you great confidence on your gating in this particular case. Even the parameters generated through the image analysis can be plotted and gated and subgated so that you can ask image and location-based questions on populations that were defined by otherwise traditional flow cytometry. In this particular case, this study was looking at levels of apoptosis in this population. Through image analysis, you can quantify the total number of fragments as a number, but there's actually a number of modalities that you can use to quantify that particular trend. So with DNA fragmentation, you would expect more independent spots as opposed to just one in this case. Here, that parameter is being plotted on the x-axis against the nuclear area. So our rates are extremely competitive. That is to say that we are way below the national average on the hourly rate. This is partly the advantage of being a large facility and a bulk volume. It's something that we work hard to maintain and are particularly proud of. So I want to point out here, as was originally mentioned, the YCC provides a very substantial copay of 10%. So through your YCC membership, you automatically pay only 90% of what non-CC members pay. A lot of this campus is not aware of this, and this has been in place for quite some time now. It's a huge advantage to, obviously, to the Cancer Center support, for which we're very thankful. Okay, I'll stop there. Answer any questions if there are any.